The world that we see today is a far crisis from what it was century ago. There has been development in the field of science from left to right. Constant research by scientists has contributed to the advancement of medical science, which has led to the discovery of so many antibodies for diseases which were once fatal. Knowing the importance of this field, the government of our country has been providing considerable financial support to many medical research institutions. Hybridoma is one such technology which the scientists have been using for years to produce antibodies. Therefore, it is important that we know everything about this technology which has been aiding antibodies production. Therefore, in this session, we shall discuss about hybridoma and hybridoma technology. We shall also discuss about the applications of hybridoma technology. Hybridoma cells are engineered to develop a desired antibody in larger amounts to develop monoclonal antibodies. By using hybridoma technology, which is a popular technique, monoclonal antibodies can be developed in specialized cells. Georges Kohler and Cesar Milstein are the two scientists who discovered this technology in the year 1975. Along with them, Niels Jerne was awarded Nobel Prize in the year 1984 for physiology and medicine. For the analysis of parasites, antigen monoclonal antibodies are so valuable and very appropriate that World Health Organization would have organized a symposium which brought in together those who have established and refined the technology and those who are currently using it or wishing to use it for the study of organism that is responsible for some of the major diseases affecting the mankind. Let us see what is hybridoma technology is. By injecting a specific antigen into a mouse, hybridoma is produced. It is believed as a hairy cell procuring the antigen specific plasma cells which produces antibody cell from the mouse's spleen, and the concomitant fusion of this cell with a myeloma cell which is a cancerous immune. Many identical daughter cells can be produced by cloning the hybrid cell which is thus produced. The immune cell product is then secreted by these daughter clones. They are called as monoclonal antibodies, as these antibodies come from only one type of cell known as the hybridoma cell. The advantage of this process is that it combines two different types of cells qualities, that is the ability to grow continually without any interruption and the production of large amounts of pure antibodies. For preparing monoclonal antibodies, HAT medium, that is hypoxanthine aminopterin thymine, is used. Laboratory animals such as mice are first exposed to an antigen in which our interest lies in isolating an antibody against it. The B cells are fused with immortalized myeloma cells once splenocytes are isolated from the mammal using polyethylene glycol or Sendai virus. This immortalized myeloma lacks the HBPRT gene, that is hypoxanthine guanine phosphoribosyl transferase gene. Fused cells are incubated in the HAT or hypoxanthine aminopterin thymidine medium. In the myeloma cells, aminopterin dies since they cannot produce the nucleotides by the salvage or de novo medium blocks the pathway that allows for nucleotide synthesis. Hence, unfused D cell die. Due to their short lifespan, unfused B cells die. Simply the B cell myeloma hybrids live, as the HGPRT gene coming from the B cells is always functional. These cells are immortal and produce antibodies. Into the multi-well plates, the incubated medium is diluted to such a level that each well comprises only one cell. For desired antibody, the supernatant in each well can be checked then. The antibodies in a well will be directed towards the same epitope since they are produced by the same B cell, and these antibodies are known as monoclonal antibodies. When a hybridoma colony is launched, it will grow continually in culture medium like RPM1 1640 with antibiotics and fetal bovine serum and produces antibodies. A rapid primary screening process is the next stage, which recognizes and selects only the hybridomas that produce antibodies of suitable specificity. This secondary enzyme labeled conjugate, the hybridoma culture supernatant and the chromogenic substrate are incubated then, and the formation of the colored product will indicate a positive hybridoma. Immunocytochemical screening can also be used alternatively. To grow the hybridomas, multi-well plates are used initially, and after the selection, they are changed into larger tissue culture flasks. For cryopreservation and supernatant, this flask maintains the well-being of the hybridomas and gives enough cells for subsequent investigations. 1 to 60 microgram per ml of the monoclonal antibody can be yielded by the culture supernatant, and it is maintained at 20 degrees Celsius or lower until needed. Let us see advancements and improvements in the hybridoma technology. To improve the production of monoclonal antibodies using hybridoma technology, considerable efforts have been made during the last 10 to 15 years. These efforts include: initially, the fusion is promoted by substituting of a chemical fusion promoter for the Sendai virus. Next, the use of myelomas that don't secrete their own antibodies and therefore they do not interfere with the required antibodies production. Next, for the antibody that produces B cells, a continuous cell line sp2/0 uses a fusion partner. For optimal growth feeder layers that consist of extra cells to feed the newly formed hybridomas are used for the hybridoma production. A most common feeder layer consists of the murine peritoneal cells, the macrophages that are derived from mouse, guinea pig, or rat or extra non-immunized spleen cells. The human fibroblasts, thymus cells or human peripheral blood monocytes. Some limitations like depletion of nutrients are present in these feeder cells that are meant for contamination and hybridoma, so that interleukin 6, IL-6 from other sources of hybridoma growth factors or HGF that are being derived from human cells were used. Let us see the purification of antibodies. Before using the monoclonal antibodies for variety purposes, they may need to be purified. Before final purification for enrichment of the antibody protein, the cultures may be subjected to cell fractionation. In Escherichia coli, in the periplasm, the antibodies may be secreted, which may be then used for enrichment of the antibody, which does the simplification of the antibodies. Instead, the antibodies may be purified from cell debris or cell homogenate obtained from the medium. With ion exchange chromatography and antigen affinity chromatography techniques, antibodies can be purified. Bulk culture of hybridoma cells through serum-free media, let us see. Culturing animal cells, serum makes a necessary component of media. Nevertheless, the use of serum leads to difficulties in the purification of antibodies. And also for large scale production of hybridoma cells, it is an expensive technology for the industrial production of the monoclonal antibodies. Due to these difficulties, serum-free media are being used increasingly for culturing the hybridoma cells. Let us now move on to the advantages of serum-free media in hybridoma cell. Because of the increased initial purity and the absence of contaminating immunoglobulin, greatly simplified purification of antibodies, decreased variability of culture medium, reduced risk of infectious agents, fewer variables for quality control or quality assurance, increased control over bioreactor conditions, potential for increased antibody secretion, low or no dependence on animals, cost effective and overall enhanced efficiency. Now let us move on to know some of the disadvantages of serum-free media in hybridoma. Not all serum-free media are applicable to all cell lines. Cells may not grow to as high densities and may be more fragile than cells in serum. Media may take longer to prepare. Let us see the cloning of MAP genes and bypassing hybridomas. Using the universal primers through polymerase chain reaction or PCR, the VH and VL genes for antibodies can be amplified. In PCR, primers by building restriction sites, cloning expression in mammalian cells or bacteria can be done directly for the amplified VH and VL genes. The hybridomas or B cells that are homogeneous or heterogeneous may be used as a raw material for PCR. Later, a variety of VH and VL genes will be amplified and combined at random to develop as many as 106 clones for the antibody genes. In a phage, these genes will be cloned and their products, particularly FAB or FAB fragments, will be screened for the antigen binding activities. In a combinatorial library from such a large number of combinations, it is very hard to recover the original pairs of V genes. For example, VHX, VLX and VHA. VLA is an original pair. By using the antigen selected B lymphocytes, the complexity may be reduced, however. Now let us see the designing and building the MOP genes. In recent years, the antigen binding sites of the antibodies have been studied in some detail, which led to the modeling of entirely new antibodies, which is sometimes for their use as enzymes. For alteration of the antibody genes and for the synthesis of entirely new genes, this modeling by computer graphics can be used. These genes could be cloned and expressed in the bacteria. The antibodies synthesized can be tested for their specificity and their affinity for the specific antigen. Now let us consider the applications of monoclonal antibodies. There are numerous use of monoclonal antibodies, and they include the diagnosis, prevention and the treatment of disease. Example, monoclonal antibodies can distinguish the subsets of B cells and T cells, which is very helpful in identifying the different types of leukemias, and also to define cell surface markers on white blood cells and other cell types. Specific monoclonal antibodies have been used. This led to cluster of differentiation series of the markers. They are often mentioned as CD markers, and they also define several hundred different cell surface components of the cells, each assigned by binding of a particular monoclonal antibody. For sorting the specific isolation by fluorescence activated cell of particular types of cells, such antibodies extremely useful. Let us see how antibodies in diagnostic histopathology method is used. Based on the expression of particular defined markers which reflects tissue or cellular genesis with the aid of monoclonal antibodies, tissues and organs can be classified. Human prostate specific antigen, placental alkaline phosphatase, and the others are organ associated antigens, and the monoclonal antibodies production against these antigens helps in determining the primary tumor's nature. In differentiating the morphologically similar lesions such as adenocarcinoma, pleural and peritoneal mesothelioma, and in the concluding of the organ or tissue origin of the undifferentiated metastasis, monoclonal antibodies are extremely useful. Selected monoclonal antibodies help in the detection of occult metastasis by immunocytological analysis of lymph nodes, bone marrow, other tissue aspirates, and other tissues. The immunohistochemical staining of bone marrow aspirates are very useful to identify occult bone marrow metastasis in the patients with evidently localized prostate cancer. The immunocytochemistry that uses tumor associated monoclonal antibodies has led to an improved ability. Only tumor associated and non-tumor specific monoclonal antibodies are used in immunocytochemistry, and that is one major drawback. Some cross reaction with normal cells can occur as a result. It is important to detect even some small quantities of the breast cancer cells in order to effectively stage the breast cancer and assesses the efficacy of purging regimens that are prior to autologous stem cell infusion. Due to their simplicity, sensitiveness, and specificity, immunohistochemical methods are ideal for this purpose. The early dissemination of malignant cells is one of the main reasons for metastatic relapse in the patients with the solid tumors. The use of monoclonal antibodies, called as MABs, specific for cytokeratins, can identify disseminated individual epithelial tumor cells in the bone marrow. By using the immunocytochemical techniques, we can avoid performing procedures that are painful, expensive, and uncomfortable to the patient. This technique can also aid to speed up the start of the appropriate treatment. Two antigens in the same smear can be detected by immunocytochemical staining. This is also another application of immunocytochemical staining. Double staining with light chain antibodies and with T and B cell markers can show the neoplastic origin of a lymphoma. To conclude, by going through this session, we got to know that hybridomas are cells that have been engineered to produce a desired antibody in large amounts, to produce monoclonal antibodies. By using a technique called as hybridoma technology, monoclonal antibodies can be synthesized in specialized cells. To improve the production of monoclonal antibodies using hybridoma technology, considerable efforts have been made during the last 10 to 15 years. We also discussed about the advantages, disadvantages of the monoclonal antibodies and the hybridoma technology. Many applications that are extremely useful were also discussed above.